Google, sir.
Good evening, all. Good evening. I cordially welcome you all for this virtual gathering. First, I invite Dr. R. Sadish Kumar, Assistant Professor, Head, UG Department of Biotechnology, to give a welcome address.
Thank you, sir. I take this privilege to welcome each and everyone joined the meet virtually. With the blessings of our former President, Dr. M. Archami, sir, I welcome our most respected Secretary and Director, Dr. C.A. Vasuki, ma'am, and I extended the welcome to our respected Principal, Dr. M. Lakshmanaswamy, sir, our Dean Academics and Department Head, Dr. S.R. Madan Sankar, sir, and our Associate Dean R&D, Dr. R. Sivaprakash, sir. And I am glad to welcome our eminent speaker, Dr. J.Jannet Vennila, Director, Self-Finance Team and Coordinator, Computational Laboratory, MCC MRF Innovation Park, Madras Christian College, Chennai. I extended my sincere welcome to all the faculty members, research scholars, and students for these wonderful occasions. Once again, I welcome everyone. Thank you.
Thank you, sir. In this juncture, I invite Dr. R. Arvind Gant, Assistant Professor, to give a brief introduction of our today's research person. Sir, please.
Thank you, sir. It is my immense pleasure to introduce our today's chief guest, Dr. J.Jannet Vennila, ma'am, Director, Self-Finance Team and Coordinator, Computational Laboratory, Madras Christian College, Chennai. She completed her MPhil and PhD degree from Avinashi Lingam University, Coimbatore. She has more than 26 years of teaching, research, and administrative experience with 43 peer-reviewed Scopus in-depth journal publications, 21 conference proceedings, 8 book chapters, 1 patent, and 1 monograph. Her research guidance includes 7 PhD, 4 MPhil, 35 MTech, MSc, and 28 BTech projects. Her current field of research interest is proteomics, phytochemistry, pivot metabolites as therapeutic agents, and network biology in cancer. She is the recipient of prestigious United Work Fellowship 2017 to 2018, where she was sponsored to attend the Summer Institute program at Harvard University, USA. In July 2017, an Asian placement at De La Salle University, Manila, Philippines during May to August 2018. She served in different administrative capacities of Karunya Institute of Technology and Science. She also served as chairman of Institutional Animal Ethics Committee and member secretary of Institutional Ethics Committee for Human Research and so on. She has vivid research exposure in abroad at Oslo University, Norway University of Toronto, Canada, Max Planck Institute of Molecular Genetics, Germany, and so on. Once again, I welcome you, ma'am, for this wonderful evening. Thank you all.
Thank you, sir. With this brief introduction, I would like to invite our research person, Dr. J. Jannet Vennila, Director, Self-Finance Stream and Coordinator, Computational Laboratory, MCC MRF Innovation Park, Madras, Christian College, Chennai, to take over the session. Madam, please.
Thank you, respected Principal, Secretary, and my close associate and long friend, Dr. Madan Shankar, and my very close associate, Dr. Satish, and my dear students. I'm extremely glad to be here. It's my honor and privilege that I could be a part of this discussion forum where I can share some few of my expertise in computational science as well as touching some biotechnology aspects as well. So I'm very happy to see my first batch of student when I started my career in 1997. and your HOD, Dr. Satish, was my first batch of student. I really cherish those moments. I'm very proud uh, to see him flourishing in career and also in academics and research, and he has rose to this level. I'm very happy about that, and I congratulate him for that. And I'm also happy my another colleague of mine, Dr. Madan Shankar, is also with the Kongunadu Arts and Science College. So I just feel it is a just yet another home away from home, seeing you all guys there in a very high position, and I'm happy about that. So this is, I think this is a students group from Department of Biotechnology. I suppose it is a UG, PG, and research scholars, right?
Yes, ma'am.
Okay. So what I'm going to discuss is not a cutting edge technology, but that will really help you people, uh, even with the basic facilities. You can think about the research and you can come with good publications or patents. That's my motivation, because I don't want to touch upon any cutting edge technology that will so we as a I mean, in Indian context, you see that universities and colleges may not have the facilities as we have in abroad, but with the limited facilities we can still do a better research. That's the motivation I wanted to give to you. Shall I share the screen?
Yes, ma'am.
Is it visible?
Yes, ma'am. It's visible. Yes, ma'am.
Yes, I just wanted to know whether the screen is moving, right?
Yes, ma'am. It's
Yes, ma'am.
moving.
Yes, ma'am.
It's
Okay.
moving.
Yes. So, although I started my career in NGP Arts and Science College, where I was a faculty in biochemistry. Later, I studied bioinformatics, and most of my research work and my postdoc work was on bioinformatics, mainly in University of Oslo, Norway, where I worked with one of the professor, and we developed a tool for microarray data analysis using Java. That was my first postdoc experience in University of Oslo, Norway. Later, in Max Planck Institute of Molecular Genetics, I worked with the system biology approach of certain cancers. And the University of Toronto, Canada, I just extended my work. So, although my career I started as a biochemistry, I have an interdisciplinary approach both in biotechnology and bioinformatics, because from after completing my postdoc, I joined as a faculty in Karunya University, Coimbatore. For the past 18 years, I was there. And recently, 9 months back, I joined as a director for the evening college and also the Computational Science Laboratory in MCC MRF Innovation Park, where I'm doing molecular docking studies, simulations, and also some NGS next generation sequencing based work. This is my brief profile. So the college where I'm just presently working has a history of only 87 years old, and it's a liberal arts and science college. Um, uh, the facilities may not be as good as you people have in Kongunadu Arts College, but it's a UGC potential of excellence. We have all facilities related to FIST facilities and bioinformatics facilities, computational science. We have a workstation and also the Schrodinger software for working with drug docking and drug designs and other things, so on. So this is the brief history of me. Before me starting the lecture, which goes like this: ribosomal inactivating proteins with medical applications. This is a very generalized topic. What I'm going to discuss with you is a generalized topic with some case studies where I have personally involved in some research activities. And this may be a real motivation for you to do some research with the basic facilities, whatever you have in hand. Before going into the slides, I just wanted to share, because you are a students, I just want to share a small story about a student. How many of you are interested to hear the story? Real story? Students? Are they all muted?
Unmute பண்ணி பேசுங்க.
Yes, you wanted to have a hear a motivational story?
Yes, ma'am.
Okay, that's good. Okay, this is about a student called Karthik Sane. Okay, so this guy or this boy was just like you. Couple of years back, he studied in Delhi public school, right? And the speciality of this child is he's totally visually challenged, so he is totally blind. Okay. And he opted to study computer science stream, physics, chemistry, maths, computer science in his 12th standard. You know how difficult is for a visually challenged child to study the science stream in 11th and 12th standard, because as a normal person we itself struggle to clear mathematics paper. But in spite of that, this boy really put in maximum effort to score 95% in class 12. Okay, so 99% in computer science, 95 in mathematics, English, physics, chemistry. So total score was 479 out of 500. And it's really a surprise, and also it's really a amazing fact that he could score 95%. And I was just reading about his his case, and he was speaking to press people a couple of years back. I have noticed this video from that. And he has an opportunity, or he was selected to do computer science engineering at Stanford University, California. Before that, in spite of him getting 95% in plus 2, he was denied twice by JEE exam, because JEE is the common entrance exam to appear for IITs, right? You all know about that. So IIT JEE exam doesn't have a provision to admit a totally visually challenged person for entrance exam. So he tried once and twice, but he couldn't be granted to even appear for the exam. So after trying hard, dejected, he applied for Stanford University. Stanford University accepted him with full scholarship. I think he would have been in second year or third year by this time to do the computer science engineering. So what I'm exactly saying is, your hard work, your perseverance, and the faith in God can really, you can live to your dreams. And I was just reading his story, and one of the press people asked him what was the real motivation behind his success. So one of the press people asked him what was the real motivation of his success. So he was saying that when I was doing 11th and 12th, at times I feel so rejected, because I have to, I have to convert the daily lessons to a format or audible, audible format that he could hear and write. So it approximately took twice or thrice effort than the normal person. So he has to work in very hard for each paper. But towards the end of the 11th standard, there was a science exhibition that was going on in a school. For that, at that time, our president, late President Dr. APJ Abdul Kalam, visited his school for the science exhibition. So as a children and the students, we know that if an influential person like president comes to a school, normally we'll stretch out our notebook for autograph. That's what this guy also, Karthik Sane, also stretched out his notebook to get an autograph from our late president, Dr. APJ Abdul Kalam. You know what he wrote in the notebook? Let me show you. Can you see the screen? Yes?
Yes, ma'am. Yes, yes, ma'am.
Yes. So our late president has written a statement. son, it is better to have a vision. That's a foresight rather than having a vision, that is eyesight. So it is better to have a vision, that is foresight, rather than having a vision, that is eyesight. So Karthik Sane said this sentence really motivated him to better, and he stands where he has to be. So your clear vision is very, very important for proceeding to either a career or a family life or your academics or your research. Always be clear in your vision and plan your milestones, plan your activity that will lead to real success. Okay. So are you all motivated by the story of Karthik Sane? Yes, really, I was motivated. You should also have been motivated by this. Okay. Now we'll come to the lecture, which says, okay, coming to the ribosomal inactivating proteins, first I need to tell you about the background of what is exactly ribosomal inactivating proteins. Before that, you all know, you have heard about the term called toxins. As a biotechnologist, you know what is toxin and toxoid. Have you heard about this? Toxin and toxoids. Yes? Okay. So plant proteins, uh, since from the time I know, plant proteins, there are many plant proteins, they are used as a toxins, either in medicine or in diagnostics or applications. Plant proteins are used as a toxins. Okay. This plant toxins, why they need to go for plant toxins is these are all natural bioactive compounds. So there are two category of plant toxins. One may be a low molecular weight, another may be a high molecular weight. When you come to the low molecular weight, we all talk about the phytochemicals present in the plants. It may be an alkaloid, or it may be a terpenoid, or it may be a tannins or glycosides. But what I am focusing is not low molecular metabolites, but high molecular weight metabolites like proteins and peptides, which may be toxic. At the more, I'll be focusing on toxic proteins. Okay. So some of the toxic proteins which is listed in plants according to Dang et al. In 2015 is, you all know about lectins, protease inhibitors, thionins, ursulins, spore-forming toxins. You would heard about that. But all the more, I'll be talking about ribosomal inactivating proteins. The term may be new to you, but this ribosomal inactivating proteins has been in the talk, has been in the medicine for a quite long time. Okay. So this ribosomal active proteins, as we know, the proteins, the structure, some of the protein structures has been elucidated. Beautiful structures are there with the two or more chains. Okay. Okay. So what are ribosomal inactivating proteins? RIBs. In general, I can give you a short term, RIBs. Okay. These enzymes possess N-glycosidase activity. Have you heard any structure with N-glycosidic bond? Maybe some of one or two of your students can unmute your audio and you can talk. Any structure with N-glycosidic bond? Have you heard about or have you come across any structure with N-glycosidic bond? What is N-glycosidic? What is glycosidic bond? First of all. Sorry for reminding you about biochemistry, but no other go. What is glycosidic bond? Yes? Satish? Okay.
Students, unmute and talk.
I think Dr. Satish has instructed the students to keep their mouth shut.
No,
Right?
no, ma'am.
Okay. So if you want to answer, because I wanted to take it as a lively, yes, don't think it is yet another class. You can feel free. Dr. Satish is not here to instruct you. You can be open, right? So N glycosidic bond. What do you mean by glycosidic bond? Is a sugar is linked to another stuff, or sugar is linked to another sugar through a bond, OH bond, O bond. You know that sugar has many OH. So through OH, it forms a bond. For example, I can say a glucose is linked to another glucose by alpha 1,4 linkage in case of your amylose. Okay, and glucose is linked to fructose by beta 1,2 linkage by in a sucrose. So these are all called glycosidic bond. But what do you mean by N-glycosidic bond? You can see in the structure of nucleic acid, nucleotide. You know that nucleotide has three basic components. One is the sugar, another is the base, another is a phosphate group. Okay, so a phosphate group, a sugar, and a base. Base may be a adenine or guanine. It may be a purine or pyrimidine. So you know that phosphate group is linked to the 3' position of the sugar, and the sugar is linked to the base through a N linkage. What is N linkage? The base gas at N at position 1. So N is linked to the OH group of position 1 in the sugar. So this you call it as N-glycosidic bond. So you know that this is the structure of any nucleotide. So a polymer of nucleotide, you call it as a nucleic acid. So any enzyme which breaks this N-glycosidic bond is called as N-glycosidase bond. So that means base is removed from a nucleotide. Okay, so the base will, when the base is removed from the nucleotide will have only the phosphate group and the sugar. So the base is removed. So obviously without the base, there is no hydrogen bonding between, you see, double helix, and everything is altered. So, you know, without the nucleic acid, without DNA, there is no mRNA. And without mRNA, we don't have any proteins. So obviously, I can say the enzyme which causes N-glycosidase activity will inhibit the protein synthesis. So this ribosomal inactivating proteins are known to be present in fungi, plants, algae, bacteria. You may ask me why. Why they have to be present is just to defense. Anything which attacks them, it will be toxic to that and prevents from the predators. And also during the stressful situation, plants produce more of this ribs. A stress may be from a predator, or a stress may be due to heat or salinity. You can see even in marine algae, when there is a stressful condition, when there is too much of tide or something else, more of RIPs are produced. So another important feature is within the same plant itself, different tissues may have the different ribosomal inactivating proteins. For example, a leaf may have a different RIP, the stem may have different RIPs, and the flower may have different RIPs, and so on and so forth. Okay. And the first identified RIPs was resin. Most of you would have heard about resin. I don't know. Resin, Tamil pathina ஆமணக்கு செடி. You would have heard about ஆமணக்கு செடி. So from the resinous communists, they isolated from the seeds of resinous communists. They isolated a very toxic substance called resin. Resin was used as a bioweapon, I think, in First or Second World War. Okay, resin is still it is a prohibited stuff in India. Okay, resin and aberrin. These are all common terms given to RIP, which has been extracted or identified in different plants. Aberrin from the abrus. Aberrin means in the rosary piece. You know that Catholic priests, they use the rosary stuff. So that also contains aberrin. So different plants, different species, different families, they have isolated and identified the ribs. The ribs may be toxic or non-toxic. Mostly it is toxic. Okay. Okay. I'll tell you how it becomes a toxic, how it acts as a inhibitor for protein synthesis. Okay, let me remind you or make you to recollect the structure of uh, or the process of protein synthesis. Okay, so if you see, for protein synthesis, you need to have a ribosome. Obviously, you know that ribosome is the site of protein synthesis. So the ribosome is the stuff which will bind to 28 RNA, bind to the factors, initiation factors. So it is the main site of protein synthesis. So the ribosome is made of two major subunits, largest subunit and smallest subunit. In case of prokaryotes, you may remember that it is made up of largest subunit is 50S, smallest subunit is 30. Yes. Okay. In the case of eukaryotes, the largest subunit is 60S and the smallest subunit is 40S. So in the 60S, you can see the other types of rRNA. Okay, like 28S rRNA, then 5S rRNA, 5.8S rRNA in the 60S subunit, while in 40S you see 18S rRNA. Okay, so the RIBs mainly attack the 28S rRNA. Okay, so the 28S rRNA, which is a part of 60S subunit, you have to remember that the smallest subunit will be there around that. Sorry, smallest subunit will be down around that. You have the another subunit sitting as a cap. Okay, so the 28S rRNA is a part of largest subunit. And when you just carefully observe the structure of 28S rRNA, it has a stem loop like structure. Okay, stem loop. Okay, stem loop. So this is the arrangement of nucleotides in the 28S RNA. So, okay, at the position 4234, at the position 4234, there is adenine. Okay, at the position 4234, there is a adenine nucleotide. Okay, adenine nucleotide means it has adenine, sugar, and the phosphate. Okay, so each nucleotide is linked by a glycosidic bond. So what is the mechanism of action of this RIP is they go and directly attack this adenine at the position 4234 and remove the base. Okay, they remove the base. So without the base, what happens is the structure or the architecture of nucleotide arrangement is disturbed. Okay, so this 28S rRNA, I can call it as at the position 4234. Without the base, it becomes headless. Okay, you imagine a person without the head, there is no use. Okay, headless. So it becomes a basic site. A basics, no, without the base. Okay, or A purine. A purine means adenine is a purine, it becomes A basic or A purine. So you see, it becomes A basic. So why this position adenine is very important, and what happens when this adenine is removed by the ribosomal inactivating proteins is, this is the main site of elongation of protein. Okay, so you know that if you recollect, I don't know, first year might not know, second year, third, you would have studied the process of transcription and translation. In translation, you have elongation factor, which usually comes in attached to CTP, GDP. So this elongation factor has to bind to the stem loop structure of the 28S RNA so that the elongation happens. So when this site is blocked because adenine is not there, the structure of this 28S RNA is distorted. No longer elongation factor can bind to that ribosomal part, and the translation is stopped or protein synthesis is stopped. So this is the mechanism of action of ribosomal inactivating proteins. Are you clear about that? Okay. So there are two major types of ribosomal inactivating protein, type 1 and type 2, based on their molecular weight and also the structure. Okay, so the type 1, usually the molecular weight is 23 to 35 kilodaltons, and it is a single polypeptide chain protein. Okay, so it has only N-glycosidase activity. The type 2 is made up of two polypeptide chain. One is the one with the N-glycosidase activity. Another one is like a lectin. What is a lectin? Lectin will attach to the sugar moiety. Okay, of another protein. If another protein is a glycoprotein, so this lectin will go and bind to that. Okay, so this type 2 is made up of two polypeptide chains which are linked by disulfide bond. Okay, it has N-glycosidase activity as well as the lectin activity. Okay, so this is a bulky one when compared to type 1 because the molecular weight is 55 to 65 kilodaltons. Okay, some of the purified RIPs, many have been purified, many have been tried. They have tried to study the structures. It is in process. These are some of the one Jatropha from Jatropha and Saphonidia. Saphonidia is some they call it a soapwort. I don't know, in the tribal people, they use the seed to create foam for them to take a bath near the river or something. There soap bottom that seed, leaves and seeds, it contains a rib called saporin. Okay, how does rib enter into the cell? Okay, suppose you consume rib either by taking seeds or pure form of rib. How does it enter into the cell? I told you there are two types of RIP, type 1 and type 2. Coming to the type 2, I told you it has made up of two chains. One is the A chain, which is a glycosidase N-glycosidase activity. Another one is the B chain with the lectin. Lectin always will go and bind to the sugar. You know, the cell surface receptors are mostly glycoprotein. So the lectin is attracted to the glycoprotein. So obviously the rib binds to the receptor, and through the process of endocytosis, it gains entry into the cells. And type 1 usually forms a pore and comes inside. So either way they come inside, the disulfide bond is broken, so the A and the B chain is broken, and you have the N-glycosidase activity domain alone remaining. So this, through the Golgi apparatus, they come to the ribosomes. Ribosomes are attached to the endoplasmic reticulum. So they damage the ribosomes by remove the purine in 28S RNA and inhibit the translation. So this is the process of transport of RIP into the cells through the process of endocytosis. You know what do you mean by endocytosis? Okay, so type 1 RIPs are many are there, but mostly you see, I told you about saporin, so plant. So they are in vitro, they have studied. Okay. Mammorica charenta is your and then the pavaka seeds. Okay, it also has. Type 2, most widely studied is your resinous, resin. Resin. Okay. So mainly applications, it has n number of applications. Mainly it has antiviral, antifungal, insecticidal. They are more toxic to tumor cells. I tell you why. Because you know that in the tumor cell, cancer cells, there are more, the protein rate of protein synthesis is very high in tumor cells. So obviously, when they are attacking the protein synthesis, obviously it is more toxic to the tumor cells. And this RIP is also linked monoclonal antibodies and directed towards the target cells, mainly tumor cells or cancer cells. So they are also used as immunotoxins. Even in plants, they are highly expressed. They are made to highly express to increase the resistance to the pest. These are all studies are there. Still, concrete application has not been described. Okay, so these are all in vitro studies have proved some type of RIPs have been used for certain cancers. Okay, for mostly saporin is used for some ovarian cancer, even in bladder cancer. MC MAP, I told you right, MAP 13 from Pavaka Mamachia is used they've tried in vitro only, liver, prostate cancer, and so on. Immunotoxins, I told you about immunotoxin. Either it can be conjugated with the chemically conjugated with the monoclonal antibody, which will direct this toxin to the tumor cells, or it can be recombinant fusions also. That can be also done. Okay. So I told you the saporin is the one which is currently undergoing work, and some of them RIPs are in the clinical phase trial also. It does not come to the market, right? So resin, I told you resin is in phase 3 trial, both for acute myeloid lymphoma and also colorectal cancer. And saporin was also in clinical trial 1 with the B-cell lymphoma and also bladder cancer. This mamodin is also tried for bladder cancer. Okay, so these are the few applications of the RIP. Okay, right now I come with my work where we work with RIP. Okay, ribosomal inactivating proteins. We used very basic facilities available in our lab. Okay, it is not. That's why I'm saying it is not cutting edge technology, but with the available resources in your lab, you can do enormous work. Where there is a will, there is a way. Okay, so this is the ICMR project. It was done by a couple of students and mainly my PhD scholar. And here we purified and characterized and studied the RIP from Ananas squamosa. Okay, this Ananas squamosa is nothing but custard apple. Yes, so from the custard apple seeds, we isolated the RIP, we purified it and characterized it and studied the all the medical applications. Okay, so this was published in 2020, recent publication in a Elsevier journal with impact factor 4.64. Okay, this is my student, and this was published. Okay, why I'm just telling you about this work is, being your PG students or a third year students or research scholars, when you work with bioinformatics, because we use some bioinformatics applications as well. When you use bioinformatics tools, always you have to rely upon the databases where already stored information is there. Okay, a very limitation we faced in this study is Ananas squamosa, the genome is not sequenced. Okay, Ananas squamosa genome is not sequenced. We don't know what is the sequence of this protein, sequence of this plant. So obviously we don't know about the genes. We don't know about the annotated function of each genes and so on. So it was a random random method we adopted, but still we were able to succeed and we were able to publish, and we are in the process of taking it further as well. Okay, so whenever you come across some hurdles saying that this is not available, this is not available, try to think it in different angle. Surely there will be a way. Okay, so this Ananas squamosa is custard apple or sugar apple. Everywhere you see it is used. Obviously, it is an edible fruit, but you should know that the seeds or leaves or fruits are used in Ayurvedic preparations to treat many disorders. Many medicinal properties have been studied, like cytotoxic, anti-diabetic, so many things. Even abortifacient activities also have been studied, mainly in vitro studies. Okay, so why actually we chose Ananas squamosa means my student, my PhD scholar, she had a farm of full of this custard apple trees, and also her earlier work was full on this, so there was no difficulty for her to get the seeds, flowers, and other things. And also she was very much interested to come with a product level based outcome. So that's why we chose this plant. And why we went for RIP is there is no previous studies which portrayed that they have not identified any RIPs from this plant. Okay, so that's the reason we went for that, and it was also easily available. Okay, and this RIP has been used as an insecticidal even in farms and pest control and other things. Okay, so we follow the basic steps of purifying a protein from the seeds, because first we studied with the leaves and seeds. We found out that seeds has more protein than leaves, and also seeds showed more activity than the leaves. So that's why we concentrated further on the seeds. So we started collecting the seeds. Approximately, we started with 200 grams of seeds. We used first to precipitate the proteins. We used ammonium sulfate precipitations, 20 to 80% gradual precipitation. Then dialysis. This was just a crude isolation. Then purification. We used two column chromatography, ion exchange column chromatography, that is CE cellulose cation exchange column chromatography, followed by the gel filtration column Safadas G50 column chromatography. So from the crude we got a subcrude. Then we went for ultrafiltration. So ultrafiltration we used a membrane 5 kD cutoff membrane. So filter out, because we know that all there is no RIP below 5 kD 5 kD. So most of them are reported above. So we used a 5 kD cutoff filter to filter the extract. So the protein which we retained was above 5 kD. Okay. Then what we did is we need to check the confirmation assay, because we say it is a RIP, but how do we exactly check the activity? Okay, this was quite cumbersome, but we were able to do that because we went for N-glycosidic activity. I'll tell you how did we do the N-glycosidase activity. For every time, I mean, whether from starting from the crude, from the extraction, every time we need to check for the biochemical parameters, whether there was a contamination or whether there is a, I mean, every time we have to see that other proteins are removed, carbohydrates are removed, nucleic acids are removed, polyphenols are removed. So we wanted to check whether the level is coming down so that we can get a pure protein. Okay, so then we did the STS page to check the molecular weight and banding and so on. Okay, once STS page was done, we I took the band and we further subjected to characterization by mass spectrometry, both LCMS mass spectrometry and ESI mass spectrometry, to analyze the molecular weight and to confirm the presence of RIPs. And of course, since this RIP doesn't have a background of any gene sequence or protein sequence, with no available genome available for anonymous commercial, we went for in silico approach just to predict how it could be. Maybe sequence analysis, sequence alignment, both the pairwise and also the multiple sequence alignment. Okay. So I told you about this confirmation assay. Confirmation assay has two steps. First is we need to isolate yeast ribosomes and also do a depurination assay. You may be curious why we are using yeast. Why not the other species? You need to know 28S rRNA is very specific to eukaryote ribosome. Okay, so yeast is the single cell eukaryotic stuff. So that's why it was easy to culture and harvest the ribosomes from the yeast. You can try with any other eukaryotic structure, but yeast it is easy to maintain, and that's why we used yeast. Okay, deep urination assay with that. Quality checking, I told you certain biochemical parameters like protein, carbohydrates, nucleic acid, polyphenols have to be done, and SDS page. Okay. So what is the I told you the first we need to isolate the ribosome from yeast and deep urination assay. So when we are doing the deep urination assay for N-glycosidase activity, I told you when there is a N-glycosidase activity, there is deep urination. That is, adenine is removed at position 4324. So that region will be without the base at 4324 position of 28S RNA, which we get it from ribosomes, will be without the base. Still the structure will be intact, but without any function. So when we add aniline, the phosphodiester bond will be broken. So the one with the A basic site will be broken. So you get two fragments. That is 28S RNA with 3027 at 5' and there will be one fragment. And at the 3' end, you have another smaller fragment. So this is the characteristic for a presence of RIP, indirectly reflecting N-glycosidase activity. Okay. So how do we grow the yeast? So yeast is grown in, most of you have done in your lab, yeast peptone dextrose medium we used, and we cultured the pellets, yeast pellets, subjected to ultracentrifugation, that is 11,000 gravity for 20 minutes. Then again sucrose gradient centrifugation, 200,000 for 4 hours, you get a ribosome. Okay, clear it, store it in minus 80 degrees until you use it. Okay. How do you do the deep urination assay? Take the ribosomes, okay, you resuspend it, and already you have purified the RIPs, right? So you mix this RIPs with the ribosomes, you divide into two half. Okay, why we need two half? One half you'll be adding aniline to separate the two fragments. Another half is used as a standard. Okay, you divide it into two halves. Then both the aniline treated and untreated RNAs, you subject to gel electrophoresis for separation of bands. You aniline treated, you'll get two fragments. Okay, so gel electrophoresis, 7 molar urea, 6% polyacrylamide gel electrophoresis, and you have to stain with ethidium bromide. Okay, just see the picture here. So this is a standard ricin. Okay, I told you ricin was used as a standard to check the RIP. You may ask me, how did you get the ricin? Ricin we imported from USA with a strict restriction, and we got it because it was used only for a research purpose. That's why we got it, and we compared with our purified RIPs from Ananas squamousa. Okay, so all the one plus means with aniline, minus means without aniline. Okay, plus is with aniline and minus is without. So for totally 4 lanes for ricin and 4 lanes for ribs. Okay, so you see here. With aniline, without aniline, with aniline, without aniline. Similar here. I told you with the presence of aniline, usually you will get 2 fragments. One at 3029 and as 27, another one is 269, because aniline cleaves at the A basic site. Okay, so you'll get 2 fragments. Okay, so see here you got 2 fragments here. Aniline untreated, you'll get only 1 band. Still 28S RNA is there, A basic site is there, and you cannot see the 2 bands. Okay, so same thing you see. This is the standard ricin, and this is the purified ribosomal inactivating proteins, what we got from Ananas squamousa. So you can see clearly there are 2 bands similar to the standard. So this proves that our sample has a RIP similar to the activity of ricin. Okay, and obviously we did all the quality check. You see gradually all the contaminating substance like protein, nucleic acids, and the polyphenol, gradually it is coming. So it is crude, this is dilase, this is ion exchange, gel filtration, gradually you can. But all the more if you see here, the carbohydrate content was not reduced as much as we expected. We were thinking why the carbohydrate content is not getting reduced exponentially when the protein and the nucleic acids and the polyphenol is reduced. Then we found out that usually carbohydrates is part of lectin domain. So it would have lectin would have carbohydrate would have bound to the lectin domain of this protein. So that's why you have the lectin, sugar content still not getting reduced. Okay, so when we did STS page, followed by that STS page, we got two different bands. Okay, usually STS page, you know that it removes the disulfide bond. Intact protein is broken into two subunits. So we got two bands with two, corresponding to two molecular weights. One is 21 kDa and another one is 28 kDa. So I told you most of these RAPs roughly approximates to 30 kDa molecular weight, single chain. So that's why we tend to take both this 21 kDa and 28 kDa protein. Okay, so we cut those bands and we digest it with the trypsin, and then we analyzed using LCMS and ESAMS. Okay, so characterization. So we for mass spectrometry we did trypsin digestion, LCMS, and for the searching the peptides we use mass code chain search. Sequence alignment. Further with the analysis, what we get from mass structure, we subject that to pairwise alignment and multiple sequence alignment to predict the function of the protein what we are extracted. Okay, so trypsin digestion. So the bands which we got from the our STS page, we dissolved in buffer, and we were careful to remove the color, decolorize it. We add iodoacetamide because without iodoacetamide we'll still remove the disulfide bonds. Then we have trypsin digestion, and after trypsin digestion, the peptides were stored in the tubes. Okay. Then this were sprayed into the electron, SIMS and LCMS, because LCMS LCMS is just like a HPLC to separate the fragments, and that is sprayed into ESIMS to get the ionized peptides. Okay, and we got different trypsin digested peptides, different peaks, and we searched in mass spot engine. Okay, so I told you two different bands we took. One is the 28 kDa protein, another one is the 28 kDa protein, peptide. That is the band what we get. So when we subject that into tryptic digestion and followed by mass spectrometry, we got 19 peaks corresponding to 19 tryptic digest, 19 peaks. So from 21 kDa, we got 19 peaks, and from 28 kDa, we got 18 peaks. Okay, what to do with this? Still, we were clueless because we don't have any protein or a gene to refer. Okay, so what we did? So what we did is we took all the sequence, we all took all the peptides, and we took all the RIPs which were previously reported, or the flowering plants and other things we reported. We took the sequences compared with the tryptic digest of our plants, and we found out approximately 14 plants corresponded to some similar to ours. But it was not exactly similar, I can say, because the percentage coverage was not 100% or 15% and so on. So near to that, we were able to find out one or two. Okay, so you see 30, 10, all corresponding to type 2. Okay, so you see type 2. So this is for 21 kDa protein. Similarly, if you see 28 kDa proteins, the maximum similarity 32 type 2 is there. Okay, and similarly you see type 2. So from this we can observe that our protein is somewhat related to type 2. That means it has made up of two chains. That's why we got two bands, and one should be a lectin-like stuff, another one should be a N-glycosidase activity. Okay. So it was also similar to type 2, but not exactly similar. You see, less than 37% or all, we can't call it as a similarity or stuff. So what we did is we went for pairwise and multiple sequence alignment. Where we took all the RIPs from flowering plants, magnoleophyte activation. We took all the so far reported plants, their RIPs, their sequence, everything we took. So the FASTA sequence of our known RIPs were matched with 19 I mean, your peptides of 21 kDa, 18 peptides of 28 kDa peptides. We got it from mascot, and we did pairwise sequence alignment, and it was searched with PIR database, and we checked for sequence similarity. So you see here, 21 kDa protein, 19 peptides. Among that, few peptides for 1, 5, 6, and other things showed 100% similarity with part of the sequence of RIP reported in Fabaceae, Viscaceae, and Phyloceae family. 28 kDa protein, 18 peptides. Some few of the peptides shows 100 similarity. So all were aligned to somewhat some RIP's activity is present in our plants, and they are all related to some RIPs present in the flowering plants listed here. So we went for multiple sequence alignment. Similarly, we did cluster W, and we found out the maximum similarity here. Okay, so here all belong to certain already known proteins like aberrin, viscomin, and ricin. So all these proteins are type 2 proteins and already reported. The structure is also available, sequences is also available. So this gave a clue to us that what we have extracted is RAP. It has a N-glycosidase activity. It is more or less similar to type 2, but type 2 RAP. It had activity, but it is very unique because it doesn't show similarity with any one of the protein, but sequence wise it has a similarity. Sequence wise it shows some similarity. How to go about this? Okay. So then later we thought, with this information, let us do some bioactivity studies to still confirm whether it can be explored further. Okay. So we, as usual, we went for in vitro studies, antimicrobial activities like agar-well diffusion method, anti-mutagenic activity, because you know that if you want to try with any anti-cancer activity, first we should know whether it has a potential to reduce the mutagenic effect of any carcinogen. So anti-mutagenic activity we did Ames test, and of course cytotoxic activity we did MTTSA. Okay. So antimicrobial activity we tried with all the nosocomial infections and also some fungi. Okay. And we were also expecting the result, but the results also came surprisingly. You see, this RAP didn't have much effect on bacteria, but it had some effect on fungi. Why so? Why so? I told you it doesn't have any influence on 23S RNA of the prokaryotic system. But in fungi, certain form of the eukaryotic systems, protozoas and other things make this. So likely you see some effect is seen. Okay. Anti-mutagenic activity. So in this anti-mutagenic activity, we used Ames test. So the strain we used is, we purchased this strain, Salmonella typhimurium TA100 strain. So what is the principle behind this is, this strain is very peculiar strain, which is customized strain, where this strain of this Salmonella typhimurium can grow, can grow only in the presence of histidine. Okay, so it requires histidine for its growth. Unless the medium is supplemented with histidine, the bacteria will not grow. Okay. So what happens here is, when there is a mutagen, this mutagen will revert back the histidine requiring strain to histidine non-requiring strain. It will mutate back. Okay, so obviously these bacteria can grow even in histidine absent medium. Okay, so when there is a mutagen is added, histidine requiring strain becomes the otherwise. Okay, so this is the principle of mutagen activity. But what is the principle of anti-mutagen activity? It stops this activity. So histidine requiring strain will always grow in histidine requiring strain. Even when you add a mutagen and an anti-mutagen, the reversion is stopped. Okay? So this is the basics or principles of anti-mutagenic activity studied by Ames test. So in our metabolic system, you know that liver cytochrome P450 is the major detoxifying agent. So we wanted to test with that as well, because that also should be used when your liver is a major detoxifying organelle, and cytochrome P450 is the major one involved in detoxification. Whenever you see mutagen or any other toxic substances involved. So in the presence of this metabolic activation system, we isolated this from the rat liver, and we used as a control as well to see how we can work in the our live system also. Okay, so the mutagen what we added was standard mutagen sodium azide. This is a known mutagen. Okay, and the strain was TA100, and we have to count the Tibetan column. Okay, so how do you calculate the percentage inhibition? What do you mean percentage inhibition? How much percentage of this anti-mutagen is inhibiting the activity of mutagen? Okay, that's what it is. So it is 1 minus T by M into 100. T is nothing but number of colonies per plate in the presence of mutagen divided by M. That is the number of colonies per plate in the positive and mutagen alone. Okay, first is the mutagen and the test sample, your RIP. Second is mutagen into 100. Okay, so if you see here, I don't want to confuse everything, but you see here the percentage of inhibition. How much of RIP is inhibiting the activity of mutagen? You see, it is far above high. In general, literature say less than 25% inhibition, no anti-mutagenic. But if it is 25 to 40%, moderate anti-mutagenic. If it is a higher above 40, it is a strong anti-mutagenic. Hello? Can I continue?
Yes, madam. Yes.
Okay.
You can proceed,
So,
ma'am.
okay. All the more you see, there is a high anti-mutagenic activity by the RAPs what we have extracted and purified. So obviously we can say if it has the anti-mutagenic activity, obviously it has an effect on anti-cancer and anti-tumor activities as well. Okay. Then we proceeded to the cytotoxic activity. Mainly we want to try with hepatocellular carcinoma. We don't want to try with many cancers, but hepatocarcinoma cell lines, and we use the control 5-fluorouracil. Why hepatocellular carcinoma? Okay, FG2 cell lines. So if you see the global scenario of hepatocellular carcinoma, that is liver cancer, is the most common malignant cancer, and it you see that more death is due to this liver cancer. And India, you see, it is in red zone. There are many cases of liver cancer. Okay, and our study, one of our previous study, which was done in 2009, I believe. So it says Annona squamosa leaves, where we studied with the leaves. The leaves can show hepatoprotective effect against the liver injury or liver cancer in mice. This was the study we carried out. So we thought we can study with the RIP which is extracted from immunoscomes, so we can just try with FG2. So okay, that's why we went for FG2 cell lines. So as usual we did here, and you see the IC50 values. IC50 value, you see it is more effective against the FG2, 11.13. You may be surprised to know that our ICIN has 30, around 30 plus. Okay, but in the FG2 cell lines. Okay, but IC50 values of purified RFP against was 11.13. So it is more harmful to the cancer cells or cancer cell lines. Again, we tried with a normal cell line, and so we found out that compared to ricin, it is more safer in the normal cells. As I told you, normal cells, the rate of protein synthesis is not as much as the tumor cells. That's why purified RIP is more effective against the cancer cell rather than the normal cells. So that's where we. So we couldn't continue further all in vivo studies with that. So we start with that, and we found out it has good activity. Still, we have to go with this because of our fund exhausted. So we were not able to. But still, I'm thinking of taking further how we can crystallizing the protein, because still the sequence is not available, so we were not able to do. But still, the protein can be crystallized. It's very difficult to crystallize the protein. I sought the help of Madras University to crystallize using another methods. If it comes so we can solve the structure, crystallize structures, and still more we can refine the study, active sites, how it binds, and other things can be done. Once it is done, probably we can think this RIP can be also used as an immunotoxin. And another MTech student also completed the study where this RIP we was found to, I told you about the lectins, right? It has two domains, two chains. One is lectin chain and other things. We found out that lectin chain is nothing but mannose binding. Mannose 6-phosphate was the one which binds. So that study is also there. So still we wanted to take it further due to time crunch and money exhausted from ICMR. We were not able to continue, but we are just doing little more things to make it more appropriate. So this is my team, entire team. This is my main scholar who worked in that. Apart from that, I have some few postdocs also. We are helping from out, and certain informatics students, both BTech and Tech scholars students were there. So this was fully supported by ICMR. And Karunya University, where I worked, they gave me all the provisions to work on that. So I told, that's why I'm saying repeatedly, with the limited facilities, we were able to come with some basic informations that we can still proceed further. Okay, so just I want to leave you with the thought that always don't think research is something beyond our imagination. So research is to see what everybody else has seen, but to think what nobody else has thought. Okay, research is just to see. All might see, all of them will see, but you have to think what other has not think and work on that angle. So that's the research. So now research is nothing a Greek or Latin word. It is a part and parcel of our life. So for all the research, you need to collaborate with each other, cooperate with each other, co-create with each other. That's where. So gone are the days where you are working in isolated pockets. So you need to come out of the cocoon. So biotechnology has to collaborate with other departments, physics, chemistry, botany, zoology, even other liberal courses. Okay, so they all can work together. Then only a research can be possible. So interdisciplinary and multidisciplinary research are the one where maximum funding is there. Interinstitutional projects are there where maximum funding is there. So as a students, try to collaborate with each other within your institution, outside the institution, cooperate with each other, co-create a space for research that will really help you out. Okay, so thank you once again. This is the innovation park I'm also taking care of, computational science laboratory, where we do some simulation work, and also we do dynamics work, and also NGS work. Thank you. Thank you for this opportunity once again. Over to you for any questions.
Thank you, ma'am. Thank you for your excellent talk. Ma'am, you have covered most of the basic techniques which we can use it in, which we are regularly are using it in our biotechnology laboratory, starting from the protein extraction, anti-cancer studies. Most of the techniques you have covered it in your presentation. Thank you, ma'am. Students, if you have any questions, you can unmute your mic and you can ask. This is a session open for discussion. Students.
Good evening, ma'am.
Good evening, Pa.
Ma'am, why do we use SDS phage over here, ma'am? We could use it. It will break the polypeptide chain, right? We could use native phage.
What is the problem here is when you are subjecting to trypsin digestion, you have to remove all the disulfide bonds, right? So in native page, we don't use SDS, right? In native page, as such you will get it. But for trypsin digestion and for followed by ESAMS, we need a I mean that subunit should be broken. That's why we use SDS page, or else we would have tried in another angle. Rather than using SDS page, you also use 2D page. Okay, so if we are using 2D page, we have to go for Maldi MS. Maldi MS. That is still more. I mean, what I can say, still more it is difficult for us to, because each and every spot and other things has to be worked out. So a better way to look into that is to go for LCMS. So LCMS, we need trypsin digested peptides. For trypsin digestion peptides, we need to go for SDS. So that's where we started with the SDS, then trypsin digestion, then LCMS, followed by ESAMS. Yes, are you clear?
Yes, ma'am. Thank you, ma'am.
No, if you're not clear, you can just go ahead. Native page also you can use, but the protein will be intact. I told you it is made of 2 subunits, right? It will be intact.
Yes,
So
ma'am.
if it is intact, it will be difficult for us to digest using trypsin.
Okay, ma'am. Got it.
Okay.
Good evening, ma'am. Thank you for the nice talk. Well, actually, I've got 2 questions. If
Yes.
I understand correctly from your talk, these rips are are confined to the seeds. So, do they have any activity in the plants? That's my first question. And my second question is you showed a clusterogram where you compared sequences. And was it a protein sequence or and if it was protein sequence, how was it done?
Yes. So I told you, sir, not exactly seeds. Leaves also contain rib. They have found out rib in seeds also, but you know that the concentration of protein is high in seeds. And that's why seed contains more rib than the rest of the tissues of the plant. So rib can be found in leaves also, stem also. Okay. So have I answered the first question?
The question is, ma'am, do they have any biological activity in the plant?
Yes, sir. So as I mentioned before, why these plants has to have rib is just to get away from the predators. Okay, so usually you have pest coming in, insects coming in, and even the animals, they coming and taking the plants. So in order to make this as a control measure, usually the plants try to express these proteins on their surface, either leaves or seeds and other thing, to prevent the predators using them. That is the main reason. And also to combat the stressful situations. Stressful situation in the sense, there are ribs which are expressed when there is a high pests in them. For example, you see certain plants, brinjal. I don't know about other plants, brinjal and other things. You see, if it is infected with pest, I have noticed that one of the study have mentioned that brinjal, if it is infested with the pest, if you analyze the seeds and the brinjal, they have expressed high amount of rib than the other conditions, normal conditions, when it is not infected with pest insects. Okay, so whenever the plant is experiencing stressful conditions, whether in pest or any other insects, or even in salinity or drought situations, they see that these proteins are expressed, and mainly ribs are expressed. So to compact that, so when these are expressed, normally it is a preventive measure of the plant to to prevent it from the pest attacking them. So that is the preventive measure. So this is preventive measure is to express the toxic proteins. So one of the toxic protein is RIP. That is the first question. Answer for first question. Is it okay, sir?
Thank you, ma'am.
And the second question you asked about this cholesterol W, right? Yes.
Yes, ma'am.
So I told you, sir, these it was multiple sequence alignment. We did it for not the protein sequence, but for the nucleotide sequence. Okay. So here, how did we get the nucleotide sequence? These sequence of the plants which are already genome is sequenced and the gene is annotated, we know which are the RIPs, which are the sequence for RIPs. Although the protein sequence, because many proteins are not solved, structure is not solved. So we can get the gene sequence from the genome. Okay, all the plants what I told you, which I mentioned in a multi-sequel element, their genome is available, and the sequence of the RIP was available. For that only we got some random sequence from tryptic digest. Okay, so from the nucleotide sequence of this tryptic digest, we converted that into the protein sequence was converted into a nucleotide sequence, and then it was made comparison. It's not a protein, but it was a protein tryptic digested was again converted into a nucleotide sequence, and then it was compared, because protein sequence was not available for most of the plants, but nucleotide sequence. So the cholesterol was done with the nucleotide sequence, not with the protein sequence. We reverted back.
Thank you, ma'am. Yeah, thank you, ma'am.
Good evening, ma'am.
Good evening,
Ma'am, the session was really informative, ma'am. Ma'am, I got a doubt, ma'am. You have done multiple sequence alignment for RIP. What is the purpose of that, ma'am? And my second question is, why have you aligned only with flowering plants? Is there any specific reason for that?
Because our Ananas squamosa come under Magnoleophyta family. Okay, so
Okay,
it
ma'am.
is a flowering plant. If you take all the plants, the information will be very huge.
Okay.
If you take all the plants, all the RIP, it will be an enormous Herculean task. So we wanted to concentrate. So we wanted to concentrate only on Magnoleophyta plants. And in the Magnoleophyta division, there is different families. So in the families, we try to get the information which are the families which of plants which are already sequenced, whether the RIPs is available. So that alone we made a multiple sequence alignment. So that will be easy for us to work only on Magnoleophyta family, because Anonaceae family, none of the genome is available.
Okay.
Anenum muricata is there, that genome is also not available. So rather we have to expand our scope, because Ananas family, none of the genome is available. So we have to expand our scope from Anona to flowering plant. That is the reason where we went for that.
Okay, ma'am. Thank you, ma'am. Thank you so much.
Ma'am,
Yes, yes.
you have told RIP will act on fungus, whether it is particularly targeting 18S RNA.
No. 18S rRNA is usually you can see only in the smallest subunit. Okay, smallest 40S subunit, but this will target only 28 RNA. So the specific stem loop structure is found only on that particular A4329 position. Adenine is present only in stem loop structure of 28S RNA, not 23S RNA. So eukaryotic system has 28S RNA, 60S subunit. Prokaryotic RNA will have 23S, which is in 50S, but it doesn't mostly affect the 23S or 40 or 30S. It is mostly affecting only 60S subunit, 28S RNA. It doesn't affect the 40S. smallest subunit. So that is the site of elongation factor binding. Because you see your cap, you have a smaller cap, among that a bigger one will, if you just carefully see the structure of this ribosomes, you can see both binding, that's why you get 3 sites, P site, A site, E site, right? If you see the in translation, you see the smallest subunit and largest subunit come with each other. It forms different site. A amino acid binding site, E site, empty site, exit site, and then peptide bond P site. 3 sites will be there, right? So the translocation from one codon and another codon, you need elongation factor to come and bind to that 60S subunit, 28S RNA. So that is the important one which will translocate the ribosome from one codon to another codon. So when this is blocked, that ribosome is stalled in the same codon, so it is not able to extend. So if it is not able to translocate it, next tRNA carrying amino acid will not come, and abruptly protein synthesis will stop that time. Yes?
Thank you, ma'am.
That's why I'm saying this RIP has more effect on insects. Insects is a eukaryotic system, and certain types of fungi which will come under eukaryotic system. It can affect eukaryotic system, but not to the extent of, it can affect prokaryotic system to a minimum extent, but not to the extent what we see in eukaryotic system. Is very abrupt in ending 28S RNA. And there are studies that this RIP can be used in the antiviral study itself. I don't know how it depurinate which nucleotide of virus. They have studied for HIV. I don't know the full mechanisms. They just studied, but full mechanism is not explored. But they have done some studies on that. But I don't know which RNA it directly concentrates, or by some other mechanisms it affects that HIV growth. I'm not pretty sure about that, but very well eukaryotic system 28S RNA at question 4329 is much explored, and it is concrete now.
Okay, ma'am. Thank you.
Ma'am, well, actually, like, no, it's really interesting. You said, like, no, these rips are like, no, are acting against insects. And nowadays, like, no, as we are going on, like, no, we are seeing a lot of usage of insecticides. And maybe these insecticides are downregulating the expression of these rips. So it will be interesting, like, no, to do a study, like, no, take fruits from an organic plant and, like, no, one that has been sprayed with insecticides, and compare, like, how much of rips are being produced, like, what is the difference between them. So I think, like, that will throw a very good light on, like, what is really happening.
Yes, sir. We conducted a study in a farm where we prepared insecticide with this Annona squamosa crude extract. We didn't go for drip exactly. Crude Annona squamosa extract we prepared from seeds, and we did that. And we found out that compared to the plants, hybrid plants, where they were exposed to pesticide and insecticide, organic plants or a wild type plant, it has high amount of this drip, and also you can see control the insecticides in a better way. That is there. And also the insecticide what we prepared using the drip was very efficient. So I've recommended it was in, I did it in touch with, in collaboration with the Tamil Nadu Agriculture University. So I have recommended them to still do much more studies and come with a formulation which can be used for insecticides. We used for brinjal. Still, I wanted to, them to explore and come with the product. I told them, and they are doing that. And another thing is, as a part of study with the same RIP, we found out that And the lectin domain is nothing but a mannose-6-phosphate binding domain. There are two types of lectins, glucose lectins and also mannose lectins. This mannose-6-phosphate binding lectins are more efficient. If you just go through the literature, mannose-6-phosphate binding lectins are more efficient. Even if you could see in lysosomes, in lysosome, protein sorting, mannose-6-phosphate lectins are more efficient in carrying the proteins from the Golgi to the lysosomes, more efficient. So here also we see more mannose-6-phosphate lectins are more efficient in handling the insects and controlling the insects, which we got it from RIP. That's an interesting feature I want to add upon.
Thank you, ma'am. Maybe probably like no, you should try to elucidate the structure of this RIP and like no, try to chemically synthesize it, and then you can come out with a product which will be like we can use it instead of the available insecticides.
Yes, sir. The main problem is of the size, sir. Actually, you see, it is I mean, putting both the subunit together, it comes around 60 kDa. Approximately 60 kDa is approximately, if I can take the protein, it may come to 250 to 300 amino acids. And we have a clue what could be the sequence, but we are not exactly, correct about that because the gene sequence is not there. So when the gene sequence is there, obviously we can express in an expression vector and we can come make the protein get expressed and we can study. But since the genome is not sequenced, gene sequence is also not available. We are not able to express it or purify it. But what I'm trying with the Madras University is to just we are given the crude from, and we are asking them whether we can standardize this method to crystallize it. Even if it is some impurities are there, that can be worked out. We can remove it when we are doing XRD. Whether it will be easy for us to crystallize, but you know that it's very difficult to purify a protein with a mega size 60 kDa protein. But they are trying. Dr. Vail Murugan from Madras University is trying to get it.
To interrupt you, like you said like you do have nucleotide sequences from your protein digest. So like from that you can design primers and try to amplify it, try to get the PCR product.
Yes, sir. That also we tried, sir, nucleotide sequence. But the problem here is again, you know that one amino acid, when we reverse it to one amino acid may have a different codon and other things. So the exact nucleotide sequence we have still not. We don't know what is the splicing process or what has happened from the nucleotide sequence to the protein, what splicing has happened or some editing has happened, or this is a glycoprotein. And you know that the characteristic of the protein also changes when the sugar is added. So what are the modification, post-translation modification happens, still we are clueless. So all these are some obstacles. Randomly, we can do something what you have suggested, but I don't think we'll get a concrete, clear picture with the nucleotide sequence what we got it, because we were we are able to assume, but we not predict very correctly, because none of them showed 100% similarity. It's all about range of 30, and so we understand that it is very unique protein, but similar to the type 2 available with other proteins RIP in other plants, but it is not similar. That we know, but the functionality might be similar, but it's very unique. Unless the genome is sequenced, unless we get the structure of the gene, it's very difficult to, then it will be just beating the bush and also randomly doing, that will not help us. So that is the thing we were not able to, because we even tried with ICMR, going with another approach, but still we thought we'll have to refine our approach, then only we can ask for further funds from ICMR. But ICMR was very much interested in this project. They were willing to give fund for second phase of study, but unless we show some results in terms of like a crystallized structure and other things, it is going to be unfruitful if you again do it in the other way. That's why once the structure is solved, it is taking time. Probably I can ask for second phase grant from ICMR. Then we can talk about, once that is that, immunotoxin and other can play a way, because um, I was very much impressed to know that it is this RIP, although its functionality is similar to ricin, its toxicity is less in the normal cells compared to the ricin, and it has more effective in the cancer cells than ricin. So it is a sure protein which can be used, which has more applications, but I don't know how to go about it, because right now we don't have any structural information and other things to concretely validate it. That is the limitations we are facing.
Thank you, ma'am.
Thank you, sir.
Good evening, ma'am.
Good evening.
Ma'am, I have a doubt, ma'am. You have did the RIPs against liver cancer cells, ma'am. Are there any chance that these RIPs can affect the normal cell metabolism or mechanisms?
Yes, I told you I told you we also compared with normal cells. Okay, normal liver cells we took, and similarly we worked with the hepatoma cells, HepG2 cells. So I told you in HepG2 cells it was 11.13, that was the IC50 values,
Okay, ma'am.
which was very less when compared to the IC50 values of ricin. So that means it is more toxic to liver cancer cells, HepG2 cells, than ricin. On the other hand, when we did in normal liver cells, we isolated from mice cells, and we did the same studies. We found out that IC50 values was high. I didn't show it because of want of time. IC50 value was higher than the uh, ricin. That means it is less toxic to the normal cells when compared to the ricin. So, although I don't know what could be the reason, maybe structural difference or something else could be there. Still, because I was not able to validate if the structure is available. Based on the structure, I would have made a structural comparison between the ricin and this, and I would have said these are the binding sites and other things. Since the structure was not available, I was not able to give concretely, oh, this might be the reason. But compared to ricin, it is more safer in normal cells and more toxic in the cancerous cells. So this is the reason I'm just projecting this rip from Anuna's sperm, which is far more better than the other one.
Thank you, ma'am.
Madam, thank you,
Yes.
madam. Thank you for your detailed explanation and covering up all the questions which we had. Madam, one question, ma'am. If it is so safe, you have told that it is more safe than ricin, then better we can conjugate with monoclonal antibody, and then we can produce immunotoxin like that and all in future.
Yes, sir. But for a conjugation purpose itself, we need to know the structure.
The structure, yeah. Yes.
Structure. Yeah,
Okay.
so that is the limitations. We don't know
Yes.
the exact
Hmm.
structure.
Oh.
So structure is available. Many things we would have thought
We can.
about.
Yes,
Yes,
thank you.
sir.
Thank you. Students, is there any questions? It seems there is no questions, ma'am. Thank you for your detailed explanation. I invite Dr. S. Dinesh Kumar, sir, Assistant Professor, to deliver a vote of thanks. Sir, unmute your mic.
Good evening to one and all. Gratitude makes sense of your past, brings peace for today, and creates a vision for tomorrow. As we express our appreciation, we must never forget that the highest appreciation is not to complete words, but by live by them. As one of the member of faculty, I take this opportunity to propose a vote of thanks to those who have directly and indirectly contributed to this program. I would like to thank our beloved Secretary Ma'am, Dr. C.A. Vasuki, for her inspiration and adorable support. I take this opportunity to extend the most sincere thanks to respected Principal, Dr. M. Lakshmanasamy, of Kongunadu Arts and Science College, for his support and cooperation. I extend my deepest thanks to the resource person, Dr. Jannet Vennila, Director, Self-Finance Team and Coordinator of Computational Laboratory, MCC MR of Innovation Park, Madras Christian College, Chennai. We are enlightened with your knowledge on ribosomal inactivating proteins and its medical application. It enlightens our ideas that extend and progress. Thank you, ma'am. I extend my grateful thanks to our Dean, Academics and HOD PG Department of Biotechnology, Dr. S.R. Madan Shankar sir, for his enormous guidance in this program. I extend my sincere gratitude to the organizing secretary, Dr. R. Satish Kumar, HOD UG Department of Biotechnology, and members of faculty from the Biotechnology Department and other sister departments for helping us out wherever and whenever possible. Finally, the wonderful audience who have turned up in such a great number. Last but not least, my warm thanks to the research scholars and students for their active participation. We thank you very much for your patience and cooperation. Thank you one and all. Be safe.
Thank you once again. Special thanks to Dr. Satish and Dr. Madan. Thank you all faculties. I was really happy to have interacted with you. Thank you for the opportunity. Keep going. God bless all your endeavors and God bless Kongunadu.
Thank you, ma'am.
Thank you.